Hey everyone, welcome to our presentation all about what is statistics and why do I need it in my ISEF project. All right, so in this short presentation, we are going to cover statistics and how it applies to your science fair project, but it requires careful setup. All right, when you are thinking about statistics, when you're thinking about analyzing your project data, you're probably envisioning graphs, charts, tables, but in order to get to that, you need to collect data and organize data well first, okay? That is the first step of all statistical analysis, okay? Once you have collected and organized your data in a meaningful way, then you are set up to have success in describing the data, analyzing trends, and potentially inferring meaning from the work. All right. So first, I need you all to think for just a minute, what is your research question or your engineering goal? Think about that for just 1 minute. All right. Most people's question or goal falls into these categories here. In a question, we're looking for the relationship between your independent variable, the thing you manipulate, and your dependent variable, the thing you measure. In the engineering goal, you're solving a problem using a particular technique or invention or solution, right? And you're going to evaluate the success of that technique, invention, or solution by measuring it with a particular tool or using comparison, right? Think for a minute, which of these categories do you fall into? What are you going to measure in your experiment? How are you going to measure your dependent variable? Or how are you going to measure the success of your device? I have come up with a hypothetical research question that's going to frame some of this conversation. My research question is, what is the relationship between frog weight and frog color in frog pond? All right, so bear with me. This is a kind of study that any student might pick up, but I think it makes a really good example for what we're going to talk about today in regards to analysis. Okay, so the first thing that you've got to do in any good experiment is identify the variables, or in an engineering project, identify the goal and the variable you're going to measure, okay? In our case, our independent variable is frog weight, and our dependent variable is frog color. I'd like to just take one aside here to discuss that our independent variable in this case is not something we're manipulating. We're not controlling frog weight, but we're looking for the impact of frog weight on frog color, right? So it's what is impacting the other. Step 2 is what kind of data are each of the variables that we're looking at, okay? Let's take a look at this handy dandy chart here. When we think about frog color and frog weight, what kinds of variables are those? Okay, so let's take frog weight for a second. Is that qualitative or is it quantitative? Qualitative versus quantitative. Frog weight might be an amount like 25 grams, right? That's numerical, right? And then let's branch off further. Is it discrete or continuous? Can you have any weight of frog, or is there only certain weights that are acceptable? In this case, we could have a frog that is 25.6 grams, 29.11.11 grams, right? That is a continuous variable. But when we think about color, let's think about color for a second. Is it quantitative or qualitative? It is definitely qualitative, right? This is a quality that we're measuring. And are these named categories or categories with implied order? Okay, color, there's not necessarily an order for color, so it's a nominal option, right? It's a nominal type of data. I would encourage you, think about the variables you're measuring, the design criteria you're going to measure. What kinds of data are they? That's first. All right, so we have identified that frog weight is continuous and frog color is nominal. Step 3, in collecting data well, how are you going to measure each of these variables? We've got frog weight, it's continuous. We've got frog color, it's nominal. So for me, I know that the easiest way to measure frog weight is using an electronic balance. I am going to simply weigh each frog when I catch them. Color is a little bit more subjective. I'm going to use visual color identification to measure each of my variables, but I may also wish to use a chart so that I have something for comparison. I may have a whole bunch of frogs in different colors and make sure that I give myself a visual scale to use. This is where you think to yourself, how am I going to measure my variables? Okay. This is the key to collecting data well. This is information that will eventually feed into your procedure. So in research or engineering projects, we design either an experiment for research or a fair test for an engineering product, right? This is what feeds your procedure, like I was just mentioning. If you design an experiment, we want each of those variables to be measured accurately and reliably. In an engineering project, we design a fair test for each criteria so that the prototype can be evaluated also accurately and reliably. You need to ask yourself these questions. When you figure out how you're going to measure each of your variables, does your experiment or fair test involve comparison? Do you have a control group? Do you have a standard to compare to? Does your experiment or fair test control as many variables as possible? How are you going to mitigate some of the confounding variables in your experiment, right? How are you going to control those third variables? How are you going to ensure that the conditions are the same every time you test your prototype? Does your experiment or fair test avoid bias, right? Is there human or operational error that might be involved in your test? Can we find a reliable measurement to replace that one? And then finally, we need to distinguish chance from real difference, right? Small things can affect the outcome of this test. Have a large sample size to eliminate the random factors that might be influencing your study. So those questions of yourself and we have to find out what are the techniques you will use to reduce confounding variables or things that interfere with your results. Some examples that come up for our frog pond example are, what if some species of frogs are easier to catch than others? That means I might be inadvertently collecting ones that are all the same color, right? What if the frogs change color over their lifespan and I'm only catching juveniles while others are adults? How do I describe the difference in multicolored frogs? For example, how do I describe a brown one with spots versus one that has a mostly green body? These are all things that I need to find ways to control in my study. There are some ways to reduce confounding variables. This is kind of a long slide, okay? So I'll make this available with the video in Google Classroom so you can read this, but there are a whole bunch of techniques that we can use to reduce the confounding variables. You just have to think about which ones apply to your situation. So here we cover restriction, matching, and randomization. I have another slide where we look at measurement and calibration, standardization, as well as replication. You can pause the video if you'd like and look through these and determine which one works best for you if you'd like, or like I said, you can use the attached slideshow. For my project, though, I'm looking at measurement and calibration, standardization, and replication. Let's take a closer look at those ones. In measurement and calibration, I'm going to ensure that the measurement instruments that I'm using are all calibrated correctly. So my electronic balance is calibrated correctly and consistent to reduce measurement and error and variability, right? I'm going to standardize my procedures and protocols to minimize variability in how the experiment is conducted. I'm going to grab the frog the same way every time. I'm going to keep it out of the water for the same amount of time. I'm going to weigh it on the balance for the same amount of time and then get it right back to its original habitat, right? That also means that I'm going to think about things like, what was the weather in the pond like that day? What sorts of predators were available? And other things like that. I'm going to try and keep as much of that the same as possible. And then lastly, I'm going to depend on replication. I'm going to replicate this experiment multiple times, maybe even in multiple ponds to ensure that the results are consistent. This is something that you should be thinking about for your project as well. All right, so we have covered collecting data, right? We described our variables, we figured out how we're going to measure our variables, and we figured out how to reduce the confounding variables in our project. Now let's think about organizing data. In step 5, we'd like you to create a sample data table that you will use to collect your measurements about your variables. Generally speaking, in research, there's one column for the sample ID, one for the independent variable, and one or more columns for the dependent variable. That could be one giant table. We'll look at some examples. Usually in engineering projects, each design criteria gets its own table. There's one column for the trial number ID, and then a second column for the results of the test. Here's an example of a data table for Frog Pond. Not only do I have my frog ID number here, hopefully I've caught far more than 4 frogs. Ideally, I'd catch 100 frogs. I have a column where I record the color of each frog and the weight in grams. That's a pretty simple table. Pretend my question is actually slightly more complicated. Maybe I want to look at the relationship between frog weight and frog color in Frog Pond. That was my original question, but also compare it to lily pad pond. If I was to organize it that way, I would add a column here for my other variable, right? Not only do I have my frog ID numbers, but I would also include the pond name. Presumably, half my frogs would be from lily pond and half of my frogs would be from frog pond. Then I would also continue identifying the color for each one and the weight in grams. Let's talk about how many frogs is enough frogs. This is also known as your sample size or your N value. This depends. The number of frogs that I would need to look at in this study depends greatly on how big the pond is. Are there lots of different types of species of pond? Is there an invasive species? Are these frogs fully grown? How many frogs are there in total? Does one frog represent the whole group of frogs? In a lot of ways, we think that one frog is the same as another. But if you were to change this and make our subjects humans, let's try this question again. Does one human represent all humans? I think we know the answer to that question. It's no. Generally speaking, the greater the sample size, the better. We usually use 30 as a rough number that can produce significant results, but generally speaking, the greater the N, the better. All right, so you've collected your data, you've organized your data in a meaningful table. Now we are set up for good statistics. In step 6, you need to consider what descriptive statistics will you use and why. I know that I am going to measure the frequency of each color of frog and the average weight of each frog by color. Let's talk about that a little bit more. Oof. All right, I went out and I collected the frog information from Frog Pond. This is all of my raw data. It goes into 3 columns here. Based on what you see here, can you draw any conclusions from this table? I don't think so. They're not in any sensible order. They're all over the place. I'm not going to be able to make any sense of this raw data. Don't be this person with this data table at the science fair, right? No one is going to be able to draw conclusions from this data. However, if I go back here and I count the number of green frogs I found in each pond, and then I take the average weight of all those frogs, that is a data table I can do something with. We are describing the data using averages, okay? So, for example, I can tell my judges or my audience that I found 13 orange frogs, 12 brown frogs, and 12 green frogs. The average weight of those frogs is listed right here, and it's all in grams, right? So I saw 13 frogs. They all weighed something different, but in general, their average weight was 20.76 grams. Now we come to the question of what graphical representation works best with your data. This handy chart, which is going to be linked in Google Classroom, is one of the best ways to identify the graph that is going to work best for you. Scroll down here. You have these questions on the side here. Question 1, 2, 3, for 5 questions on the side, and it asks you something about your data. If the answer is yes to this question, one of these graphs over here may apply to you, okay? On the next slides, I have broken out each of these different questions. Does your question ask about the variability of a group of data points? In our case, kind of. Does your data compare 2 or more groups? Oh, wait, that one's closer, right? We're looking at different colors of frogs and their average weights, right? You may also consider, does your data ask how populations are related to each other? Did you measure something over time? We didn't, but you may have. Does your data ask how a total is split between subgroups or subpopulations? That one doesn't apply to us, but it might apply to you, okay? This is another alternative to the chart. Sometimes some people prefer the color one. Some people prefer this one over here. It's completely up to you. Both of them are linked in the Google Classroom. But the idea is that we are comparing 2 or more groups, which means these are the graphs that are available to us. Dot plots with bars, box and whiskers. We've got a lot of options here. So let's see what I put together. In our group comparison, we are specifically looking at color of frog, right? We're looking at green, orange, brown frogs, and this is the number of each frogs found at that location. Excuse me. Here we've got a nice clear bar graph that shows how many frogs were caught at each location and what color they were. Here we have another bar graph that looks at the average weight of each frog, right? And you can see here that the averages are included on this graph. We've got both of our axes labeled, but a bar graph is definitely a great choice. Now, I would say this is a great choice, but it's not the only choice, right? Back in that table we were looking at, they had more than one graph option. Let's try something different here. This is a box and whiskers plot, okay? So we've got our green frogs, our orange frogs, and our brown frogs. And we've got a box and whisker plot, okay? The box and whisker plot is a much more powerful display of data because not only is it showing you the average, this middle line here, this is the average for each color. That corresponds very well with this graph right here. That's the same levels that you're seeing at the top of these bars, okay? But this also shows you the maximum size of each frog, the minimum size of each frog of that color, and then this is the first and the third quartile. If you need a refresher on how box and whiskers plots work, I'd be happy to provide one. Okay, there's lots of resources on the internet that can help you with that. But this tells you so much more about the distribution of the data, right? Now I can see that a lot of the frogs were really similar in size, right? They had kind of similar maximums and very similar minimums, even though it looks like the averages are different. More detailed representation of the data means better interpretations. This is a great way to present your data. All right, so when you are describing your data, you want to consider what graphical representation works best for you, okay? Bar graph in our case was a great choice, but a box and whisker plot was even better. Generally speaking, you have one graph per variable or criteria. Sometimes you can combine them, but generally speaking, it's one per graph. Okay, let's consider a couple of hypothetical graphs. The relationship between frog length, that's over here on the y-axis, and frog mass. I know that wasn't part of our original research question, but these are two numerical values that we are trying to relate, right? These two variables would be described using a scatter plot, and we've put a line of best fit in here to describe that data. Notice the line of best fit doesn't hit all of the points. It just shows you, generally speaking, the middle point of all of those random scatter points. Here's another graph. So this time we're looking at length and mass, but we're also sorting by color, okay? If we put all three variables into one graph, it can become really messy and it's hard to interpret, right? I can see the color differences between these graphs, but there's a lot of overlap, right? We could use a scatter plot, or we could use a stacked scatter plot. Sometimes this can be helpful. Don't forget to ask yourself, hey, can I see what's going on in this graph? You can use your common sense and say, this is getting very crowded. This is very crowded to see. I'm going to create a graph that's easier to look at. Okay? Okay. So you've described your data using your averages. Sometimes we've used different graphs, box and whiskers plots. Sometimes you're using pie charts or other options, but you've got that handy guide and that question tool so that you can figure out which graph applies to you. Now it's time to analyze your trends, okay? And in analyzing your trends, you're looking at the intersection of descriptive statistics and inferential statistics, okay? In descriptive statistics, we're describing the shape of the data, the mean, the standard deviation. If you don't know what that is, don't worry, we'll get there. And then in inferential statistics, we're determining whether the effects that you saw were real or statistically significant between the groups, right? We're trying to find out if that difference you see is in fact due to the independent variable. So let's talk about something here. Let's talk about representative samples, okay? Let's pretend this is my population of frogs in Frog Pond, and I managed to go out there and catch these ones. Is this little foursome representative of the whole population? Look at the size of those frogs. Have I grabbed a representative population? I think the answer is no. In here, this is the whole population. I had 2 big frogs and a whole bunch of little ones. Here, I grabbed 3 big frogs and 1 tiny one, right? I think that our sample is not representative. But we don't have to guess because math will help us figure this out. I'm sure that's the answer that you all wanted to hear, okay? Let's pretend that these are all the frogs that I sampled. I've got their weights labeled right here. And if we lined up all these frogs by size, we would expect a few little guys, a few big ones, and then a bunch of ones that are pretty medium, okay? Right? That's what we would expect because we've got a whole bunch of frogs that are kind of medium size and then a few extremes. For example, here we go. So over here, we've got our little frogs. Over here, we've got our giant frogs. And then we have a whole bunch that are somewhere in the middle here. In fact, you could put a curve over this, right? This is what we call normal distribution. There is one peak in the middle of the data where most of the organisms fall, okay? Let's look at that curve actually a little bit more closely, okay? If the curve is normal, then it peaks in the middle, and this is your average, right? The distance from the average is measured in standard deviations or sigmas, okay? So if you're really, really big, you might be 1 standard deviation, 2 standard deviations, or even 3 standard deviations from the average. Or if you're really small, you might be -1 standard deviations away, -2 or -3, okay? You can actually calculate the standard deviation for yourself, and we did that for these organisms, but we're not going to do that today, okay? Instead, let's do our comparison again where we're looking at Frog Pond and Lily Pad Pond, okay? Here's the frogs I collected there. Here's the frogs from Lily Pad. Let's find out if this data is normally distributed. Calculate the standard deviation. And here we go. This is Frog Pond, okay? Frog Pond, we've got our little frogs down here. We've got our big frog up here. And then what do you know? Most of the frogs fall into the middle. This is a normal distribution. One big peak in the middle, and then a couple of outliers, okay? But here's Lily Pad Pond, right? Lily Pad Pond had a whole bunch of little ones, and then a couple of big ones, but not really anything in the middle. That means that we had abnormal distribution. If your data is normally distributed, that's great. That means there's a whole bunch of statistical tests that we can do on it. If your data is not normally distributed, then it changes what statistical tests are available to you. And generally speaking, there's fewer options. But This information, if you can answer this question, it will help you and your statistics advisor choose the right inferential statistic for you. Okay, here we are. We analyzed our trends. We checked to see if our data was normal. Now it's time to infer meaning. Inferring meaning comes from inferential statistics. Inferring, inferential. Those are the same on purpose. Statistical tests show whether relationships or differences are real and not just something you would get by random sampling when there is no real difference. Okay? All projects will have descriptive statistics. Many projects will have inferential statistics. Being able to explain why you don't need inferential statistics is just as important as explaining which ones you did choose. Here's the thing. We know that through your experience in science and math, you can probably get through mean, median, and mode, and maybe even making a graph completely on your own. We totally support you, and we're here to just help make sure that you're doing things the way that is scientifically supported. However, most of you have not gotten to inferential statistics yet. That comes up more in the AP Stats curriculum, and we know that a lot of students have not covered that yet. So each of you will get a one-on-one appointment with either one of the ISEF coordinators or one of our statistics advisors. One of the things that the statistics advisors will ask you when you sit down for your very first statistics advising appointment is, are you exploring a proportion or how data are distributed in categories? Are you looking at a change over time? Are you looking at the variability in data? Are you comparing groups of data? Or are you looking at a relationship between two numeric values? Do you remember which one we were doing with Frog Pond? We were comparing groups, okay? Each of these questions inspires a different type of inferential test. No expectation that you would know what these tests are, but this is what you can bring to your statistics advisor and say, I think this is my kind of data, and I think this is the test that goes with it, okay? So we've got, for the proportions, we suggest these tests. For change over time, we suggest a test called an ANOVA. For variability, we suggest a test of normality. For comparing groups, hey, that's us. We recommend something called a t-test or potentially an ANOVA. If you're looking for a relationship, we look at a linear regression or an ANCOVA. Whatever test you think is right for you, your statistics advisor will confirm that that is the case with you. They'll ask you about your representative samples, they'll talk to you about your experimental design, and then they will actually help you conduct the test. Most of these tests probably will take place in either Google Sheets or Microsoft Excel, and we will be there to help you every step of the way, right? Considering these options in advance will help you tremendously. So there's one final concept that I want to put out there just so that you know what's coming. We have covered organizing your data and collecting your data well. We've talked about graphs that are great options for you, and we've provided a choice chart so you can figure out which graph makes sense for your data. Then we've started talking about inferential statistics. Here's the thing. Every single one of those inferential tests that we just looked at briefly are going to result in something called a p-value. The p-value is a probability, okay? It is the likelihood that the difference you see between your groups is real. Let's look at that on a graph, okay? A p-value describes the probability that you would actually get your results, okay? Most of your data is going to fall in here. Think of that like your control group. But your treatment group might fall out here. A p-value that is very, very, very low means that the difference between your observed, the difference between most of your data and your treatment group, that means that it is statistically significant. All right? Your ISEF coordinators and your statistics advisors will help you figure out what this means for your specific project. We'll be there every step of the way to help you interpret your p-value. But for now, just know that's coming. It's a probability that your results are real. All right. So Mr. Shower is our ISEF statistics advisor. He serves Wilsonville and Riverside. And then Mr. Smith serves Westland High School. They will be available by appointment to help you figure out appropriate graphics, analysis tools, and how to communicate your findings. You can find appointment information attached to this link here in a couple weeks. As of right now, Mr. Smith and Mr. Schauer are both available via email for statistics questions, and so are your ISEF coordinators. Thanks for tuning in. Have a great day.